Good morning, everybody. My name is Doug Flanzer. I'm the CTO of System1 Biosciences here in San Francisco. I hope during this time you're all healthy and sane as we shelter in place. So let me tell you a little bit about System1 and our mission to cure brain disease using humanity's best science and how we used AWS to do that. And in fact, how I built the wrong thing in AWS and then built the right thing. So hopefully today you'll get some practical knowledge on the types of decisions you can make to try to make tech stacks work for the life sciences industry. And let's begin by telling you a little bit about today's presentation. So in part one of this presentation, I'll talk about System1 Biosciences and our unique human model called the patient-derived organoid, our approach to technology-driven discovery, and our ORCA, or organoid culture and assay platform. And then in the second part of this presentation, I'll tell you about our journey on AWS and how we used architecture to meet the challenges of our company, how we built the wrong thing to begin with, and then adapted to build something we love and we're super happy with. And then I'll make sure to give credit to the people on my team who made this all possible. So let's go. So the first thing is, who are we? Well, System1 Biosciences is a neurotherapeutics company based in San Francisco. And our goal is to combine human models of brain disease with automation to scale our biology to the level at which we can use machine learning to find solutions to treat neurodevelopmental, psychiatric, and neurodegenerative disorders. And in order to do that, we've really had to bring together a very diverse team of scientists and technologists. So we have PhD level scientists in stem cell, organoid, and neurobiology. We have software and automation engineers who work on our platform. And we have a team of computational neuroscientists and machine learning scientists who help us analyze the data. And all three of those teams really have to work in concert to accomplish what we're trying to do. And in order to facilitate discovery in that way, we've built a technology-enabled platform that we call ORCA, which is based on the patient-derived organoid. I'll talk a little bit about that in a further slide. And this platform facilitates deep phenotyping and drug discovery on the diseases that we care about. So why build this company, right? What's the problem? Well, the first thing is there's a huge, high unmet need in these types of drugs. Indirectly, brain diseases are the #1 cause of morbidity in the United States, accounting for over $500 billion in economic burden. And yet there's really very few drugs to treat the disease. And the primary reason is poor non-predictive clinical models. So here you can see in this graph that the likelihood of approval from phase 1 for neurology and psychiatry drugs is among the lowest by disease area. These diseases have a huge genetic complexity, and so we really have an incomplete understanding and inability to properly model the genetic drivers of many of these diseases. And also, we have impoverished models. So biochemical models or cellular models or even rodent models really fail to express the unique drivers for human disease, right? These are diseases of perception and cognition, and it's very complex. The whole purpose of preclinical models and studies is to predict which drugs will be effective in human patients. So the foundation of our discovery engine is the patient-derived organoid. We call that a PDO. And it's the most representative model of the human brain ever developed. So let's talk about PDOs. So PDOs are novel, human in vitro models of disease. Right? Essentially, it's living, organized human brain tissue grown from patient-derived stem cells. And it really unlocks a deeper understanding of brain disease. And the reason why we feel like this is the best model is, one, it has preserved patient genetics, right? It has the unique cell type diversity that we expect in a human brain. And it has aspects of the tissue architecture that we expect to accompany that, as well as having neural network activity. And there's really just no other way to get access to living human brain tissue for drug discovery. The next slide, I think, is really interesting because it shows you a glimpse of an organoid. And these organoids have complex endogenous neural network activity. So this is an example where you'll see Each blinking point is a neuron communicating with other neurons in the network. And this type of activity is essentially what's happening in every person's brain, right? Information processing. And we feel like that's critical to understanding diseases, as I said before, of cognition and perception. So this is the only type of model where you can really get all of those features in concert. So in order to do this, we've had to take a technology-driven approach. And what's our solution to this problem, right? One is using this complex in vitro model of human brain disease, and that is in the lab, right? So these stem cell-derived PDOs capture the disease genetics that we care about and the tissue architecture and cell type diversity. Second is to be able to do this at scale, right? To be able to use automation and software infrastructure to support our organoid culture and then the phenotyping and drug discovery pipeline. And the third is to use machine learning and advanced analytics in the cloud. And these comprehensive analytics sort of drive our preclinical drug discovery from target ID to other aspects of research. So you could kind of think of these pillars as being in the lab, driven from the cloud, and working in the cloud. And so this vertical integration of our technology stack is really the approach that System1 thinks is going to be successful as we take advantage of the most modern tools to study complex biology. And you might ask, like, how did we end up being a cloud company when essentially we're a biology company? I think that's a good question. So, first of all, we're a set of people working on novel biology, and we have a culture focused on building new things. And the cloud is really a perfect place for us to flex that builder or maker muscle that all of our employees sort of have. Also, we're a small company and we want to remain right-sized, but still have access to huge amounts of burstable computing power. So that's a good fit also. I mean, we generally see technology and computing as part of biotech's future, and taking advantage of this sort of best-in-class products and services from different cloud vendors is a way to accelerate our discovery engine. So it's a tool, right? We could do all of this by hand, but it's about going fast. Walking is great, a bicycle is better, a race car is best. So, that's the way we kind of think about it. So what have we built? We've built the Orca platform, which really we use to try to unlock the value of patient-derived organoids to drive drug discovery. Let's revisit those three pillars that I just mentioned, right? We've tried to systematize every part of the process by building, modular automated infrastructure that's really driven from experiments planned in our cloud-based control system. And this allows us to operate at a scale of tens of thousands of organoids in the pipeline at any time. Data that's collected from the assays that are then driven through our pipeline, and that facilitates a central location for data management, quality control, and processing at scale, right? And having that data in a central location at scale means that we can have machine learning and analytic engines that work to identify or measure disease signature in our samples or drug effects, target identification and validation. What is it? You might ask sort of what it looks like. And here's a way to sort of visualize it to the end user. And you can see here a few different visual representations of our platform. As I said before, it drives both our automation and our data pipelines. So in the top half of the screen, you can sort of see some screens that represent the control systems aspect of this platform. The management of our biorepositories, using the tools to plan experiments, and then having those experiments automatically translated to control automation. And below, you can see different visualizations of some of the machine learning and artificial intelligence pipelines that we use. So essentially, like, a central claim of the System1 thesis is that the signatures of disease are likely to be multimodal in nature. And we have to use a suite of techniques to phenotype the different specimens that we're studying to generate a range of approach that might be tailored for different data modalities. So let me be more specific. For example, on the left, you see we use bright field imaging to examine gross morphology of an organoid over time. The large size of the data set sort of makes it a good fit for deep learning. and for applications of deep learning like semantic segmentation, anomaly detection, or similarity scoring. We use immunofluorescent imaging to permit us to sort of examine the cellular organization and macrostructures within the organoid. For example, the presence of budded ventricular zones that compose here, as you can see, like radially organized progenitor cells. And we do tissue classification there to understand exactly what we have in our organoids. Calcium imaging, enables us to interrogate the dynamics of neural activity in the organoid. And high resolution videos of this over time are processed through our pipeline. And we sort of do disease phenotyping at various levels of feature extraction. So, it could be different levels of complexity, like examining the frames of the video directly using deep learning, or using traces inferred from the spikes of each neuron firing, or from summary statistics of the entire neural population, like quantities that describe the network. And then transcriptomics, and we use single-cell RNA sequencing to profile the gene expression of individual cells. And this sort of makes identification of cell type possible using both unsupervised and supervised learning approaches. And the cellular identity can be used for phenotyping, or it can form the basis of looking at differential expression for target identification. So a common thread of all this is multiple modalities, the right process, the right tools for each job, and hopefully that pushes us down the road to be able to discover new therapies. So with that, I'd like to move on to discussing how AWS fits into this journey. And anybody who's built on AWS can kind of get my insider joke here of, it's very, very common to start with something that doesn't work at all and then evolve towards the right thing. And the way I like to start telling this story is, if you're thinking about technology challenges, I think you're sort of thinking about maybe in the wrong way. So companies or organizations have challenges, right? And technology is a tool to help those challenges. So instead of thinking about technology challenges, let's just think about the general challenges that any company in life sciences that's sort of moving into the data-rich regime might encounter. So first of all, there's cultural problems, right? Biologists that are trained in a very specific way are now being asked to use specific high technology tools for data entry and analysis. And to make your company successful, you need these tools to be adopted to be effective. So any new tools you built have to beat existing ones, or you're going to face a lot of culture challenge. You have a geography challenge, which is you have people and biologists and research associates and scientists and robots and assays that occur in the lab. But the cloud, as my father would like to say, is in the sky. It's a good joke for him. We have to bridge this cross-platform old school device hardware with modern methods, right? And that can be really challenging. And we must be able to reliably move our data between the collection instruments and the cloud. We have a problem of scale, right? So the best thing for your company is to be super successful, which of course drives ever greater data volumes and higher throughput needs, right? So it's an ongoing problem of how to transfer and process your data as it grows, and this increase creates a real focus on choosing the right architecture to meet those challenges. That's of key importance. And also, if you have a small team, you sort of need to know how to get in front of the DevOps challenges of managing that scale. And finally, there's cost. So when you found a company, I think there's a lot of pressure to say, well, anything we ever do, let's keep everything forever, and let's get results as soon as possible. And let's make sure we can redo any assays studies that we need to do over time. And all of those goals, which are great goals, like really stress the budget. These cost problems tend to sneak up on you. And of course, those of us who began our careers as software engineers are still software engineers. That cost is like the last thing you want to think about. So let's talk about our architecture that we've built and how we've sort of thought about each one of these challenges and built architecture to meet it. The geography or the culture challenge, excuse me. We'll start with the culture challenge. So we tried to meet the culture challenge through a quadrant of our architecture that I call the control systems, right? Essentially, we've got to build something modern and fresh and fast and responsive that our biologists are going to use and they're going to get addicted to and they're going to love using. So what you see here is something akin to like a very modern web framework, right? We're using React on the front end with Node on the back end, and we've deployed Node in Docker on Fargate, which sort of manages our upscaling and downscaling for us and has high reliability in the way that we use it. And the API between the front and back, we've chosen GraphQL. So, you can see some screenshots of the front end of part of our control system, which is a modern web app that really gets our biologists sort of excited and addicted to using it in a secure way over both, at their desk, over mobile, when they're working at home, and that really has helped our company be successful in this really challenging time. So modern UX can really help the culture problem, and deploying on the tools that are used for modern web services really have, helped us turn the corner there. The next quadrant that meets the challenge is the geography challenge. So we talked a little about automation, and while I'm talking, I'll have robots roboting in the background so you can see a little bit of what we're talking about. But, from that control system where biologists are planning experiments, we use web services to drive the automation. So web services are connected to our robotic infrastructure, and they execute in the lab, but are driven from the cloud. The assay data that we have is written to our SAN, storage area network, and we in the lab have a VMware cluster that virtualizes the infrastructure that's controlling our automation. Like other people, we use Windows where we have to, but Docker where we can. So we really like Docker. We unify our log layer using an open source system called FluentD, and that's been a real successful way to get all of our instruments logging to the same place and pushing those logs to the cloud for sort of collation and monitoring. We use a mix of in-house Python tools and vendor systems that are off the shelf to control robots and scopes. I'd say we like API, we don't like clicking buttons. And then we use a product I'll talk a little bit more later about called AWS Data Sync to move the data that comes off of our assay system to the cloud. The scale challenge we meet by deploying a number of pipelines, essentially one pipeline per data modality to the cloud. We use a data sync to push data from the lab to the cloud. And as soon as it hits S3, we use S3 triggers to kick off processes that go downstream. We actually have an interesting intermediate step of putting everything into a queue, which I think has been a good practice to make sure we have a good place for stopping and starting our pipelines, to make pipelines easier to rerun. And one of the things we found is that the SQS triggers that kick off Lambda processes that start our pipelines are highly reliable. The S3 triggers are also very, very, very reliable, but you do also need to build infrastructure because a fraction of those triggers don't actually trigger, and you may miss data that's pushed up. We tend to use a database to keep track of our status, and then we use a combination of tools like CloudFormation, GitHub, and CircleCI for deploy and testing of the data pipelines. Finally, the cost challenge, right? So in order to do the heavy lifting, we use containers that are stored in ECR to run Docker over AWS Batch. AWS Batch has both spot compute environments and not spot compute environments, and we use both spot and not. Spot has a different level of pricing that can help you bring cost controls down, but it also comes with the downside that the instances can be yanked away from you. And that can be painful during long-running processes unless you've architected them to be able to restart at intermediate time points. We write our pipeline output to both S3 and to database. We use the Aurora RDS product. The size of our jobs varies hugely. So sometimes it takes 5 seconds to run a job, and sometimes it takes hours or even a whole day to run algorithms over GPUs. So everything scales and costs differently, but unifying our pattern has really paid dividends in our ability to sort of investigate and debug the issues, as well as onboarding new people and new pipelines. So, this sort of one size fits all, it's not technically true, but like it really helps your organization. And so I think we've gotten a lot of mileage on that. So I think it helps to think about this architecture as a loop, right? So humans use the control systems to create experiments that drive the automation. The automation creates data that's pushed to the pipeline. The pipeline kicks off processes that run the compute environments, which create data that the humans look at to plan the next experiments. So I think it's kind of a unique way to think about the problem, meeting different challenges and building architecture that address each one of those challenges. So in the final, handful of slides in my presentation, I really want to talk about, what we've learned building AWS and how I think it's very normal to build the wrong thing and then evolve and build the right thing. And in this blue text, I've said now with tips, I think it's great if you can come to my presentation and bring some concrete practical advice away. So I've tried to call that out where I could. So one thing that's really interesting for life sciences is, big data starts as small data. And the analysis pipelines tend to grow ever more demanding over time. So in the early days of our company, we used Lambda. I mean, Lambda works great. It's easy to deploy and it gives you massive parallel scale for lightweight processing. But, as the analysis packages that you're using get bigger, like scikit-learn or having to compile assets for OpenCV, we started putting effort into making packages smaller or compiling, and that sort of felt like the wrong thing to do. So we started moving towards containers, right? And we tried step functions, and step functions of it allowed us to take a hybrid approach of using existing Lambda with containers to make workflow and provide different levels of coordination. Step functions also worked, but it was a little harder to try to figure out what was going on inside and debug at the time. And as we created a hybrid solution, you inherit sort of the downside of both pieces or multiple pieces of your hybridization. So in our next generation, we moved to AWS Batch, where we found that we just fully moved to containers, regardless of complexity or size. And this unification on a single pattern really helped us. And the branching logic that maybe used to be at a higher level in step functions was moved inside the container, and essentially it was just easier to operate. So I think that was a great decision. And in the future, we want to look at broader, multiple container orchestration tools that for our longer distributed workflows like bioinformatics, and we may look at something like Nextflow. But existing solutions may not last. So just understand from the beginning that your stack will evolve and change course, and don't try too hard to fight it. My next slide is, fourth time's a charm. I think a lot of us have been here before. So, getting the files from the assay devices in our lab to AWS reliably and quickly, that's a challenge, right? And in the early days, we just had homegrown scripting that monitored local file systems and just copied it up to the cloud. And as we grew in scale and we wanted faster syncing, this became problematic. And the first thing we tried was a product called AWS Storage Gateway Appliance, which I think in many ways just has a very beautiful story, which is buying hardware that automatically syncs between your on-premise environment and the cloud. It sort of seemed ideal. For us, it had a limited compatibility with the way that we use S3 object triggers. So we were using Samba to write to our on-premise side of the storage gateway. And essentially, that was causing double uploads of every file. And we have very large files, and those files trigger pipeline processes. So we were eating up a lot of bandwidth and paying for it, and then triggering processes twice. It actually took us a while to figure this out. But many tickets later, we know this to be true, and we moved on to a different product called AWS Data Sync. So AWS Data Sync is more of a virtualized transfer service, and I think it's kind of new, and so being an early adopter and power user, of certain services in the cloud certainly is not without hiccups, but it has excellent transfer efficiency. And the support we've gotten from their product team on our Power Unique use case has been really helpful. There's no CloudFormation support for it yet, which I think you'll find sort of on a couple of AWS technologies as they rolled out. CloudFormation may or may not come right away. And so we're eagerly waiting for that because that's a big part of our stack. In the future, we're investigating, can we use this over Direct Connect, which would give us even better bandwidth and convenience. So my takeaway here is, sometimes it's tough to find the right tool for the job, and things that seem like a perfect fit may not be a perfect fit, or they may not be quite ready for prime time, and you've got to keep digging. DevOps is a team sport. So how do you maintain velocity without bottlenecks and specialist hires? So one is you might have engineers that are saying things on the left-hand side of the screen, and your job is to convince them things that are on the right-hand side of the screen. So just writing application code, instead of both doing application code and development operations, is not great. The modern way is for everyone to be able to do this. If you're building a cloud company, you need to empower everyone to understand and repair the stack. So configuration as code creates documentation and flexibility, and using CloudFormation to deploy our stack has been one of the best decisions of our organization. It allows you to do logical grouping of substacks. It allows you to single-click deploy test environments for each of your developers. It allows to hook in continuous integration. So I think it's a great decision. And the other thing is, I can't work the cloud because it's a mystery. You have to have instrumentation and logging as part of your design, right? Connect errors to monitoring and alerting. We use products like Slack and Sentry to tell us what's going on inside. Just a couple more slides, so bear with me. Another gotcha is it costs what? So looking at microscopy data, if you're in the life sciences, is a real interesting example of this. So it yields very big and fast moving data. So even if you had a single microscope like we have, taking videos at 10 frames per second, you're taking 50 or 60 of those a day, right? You're all of a sudden spending thousands of dollars a week, hundred thousands of dollars a year on storage in S3, and you're paying a lot to move those files between different life cycles if you're storing individual files. So understanding how lifecycle management works in S3, knowing the cutoffs for how long you have to be in certain buckets before you can move them, otherwise you'll pay a penalty fee. Using containers of files instead of individual files to avoid per file costs. Like, do the math so you don't get surprised, right? The default choices you'll get when you deploy a stack don't fit your budget needs. So take a look at, and talk with your systems architects or your business contacts at AWS to understand costs and plan for it. Finally, build the tools that you need to defeat the magic of bugs and discrepancies that happen inside your stack. So some of the things that we've done is build a tracking system for all our jobs, right? Like some AWS services will clean out history and you'll lose access to those identifiers if you're not tracking them yourselves. We've stored jobs, all records of jobs, in a way such that it's rerunnable, and we've invested in the code that makes jobs rerunnable. And we've built tools to rerun jobs in an interactive manner. So, runtime debugging of our cloud containers has been critically helpful to making sure we have the right tooling to understand what happened and make the stack better as we go. So invest in those tools and not just the products. And finally, my final slide is, hey, don't forget, it's an art, not a science. There are many solutions to your problems. I think I joke with my team, we call this alphabet soup. But a good example is if you need to run containers in the cloud, you can run them on batch or Fargate or Kubernetes or deep learning containers. Or if you need to queue or stream information, there's a variety of things you can do. And no systems architect can come and tell you exactly which one is right. Some of them have overlapping functionality. And it's really up to you to try them and find the right fit. There's lots of decision points for finding the right fit, but if you had to bank on one, I mean, ease of use is a great rule of thumb. Like some of them are just harder to use than others, and that's a burden and a barrier. And when there's overlapping functionality, start with the one that's easiest, and evolve from there, because you'll understand better the choices that you're making. So that was a lot. That was a lot to cram in. I'm surprised. I really hope that you took some tips away from that, and that it felt like we've built something really interesting. As we continue our journey in finding therapeutics for some of the world's, toughest neurodegenerative and neuropsychiatric disorders, I hope that you'll keep your eye on System1. For more information, feel free to reach out to me. And I want to give special thanks to the platform team, Andy Lash, Chili Johnson, and Anthony Boshart, for really making all of this possible. It's been a great ride so far, and we look forward to more success in the future. Stay safe, everybody, and thanks for your time.